Today we'll be talking about DNA replication. Right here, as you can see, I have a diagram of a DNA strand, which is two backbones, which are the two waves swerving into each other, and rungs, which are the straight lines connecting the two backbones. DNA is made up of the monomers called nucleotides, which are the sugar, phosphate, and bases here. Right here is the sugar, and right here is the phosphate, and these four are the bases. Right here is the adenine, and then this is the thymine, guanine, and cytosine. These are the four bases which make up the DNA. The structure of the DNA strand is antiparallel. The sugar-phosphate bonds, which are right here, is a very strong bond. The hydrogen bonds, which are right here, are weaker bonds. These weak bonds are important because it allows the helicase to separate the DNA strands, which allows the SSB protein to hold apart the backbones, which allows the primase to lead the polymer. The primase leads the polymerase, which builds, and then the ligase glues what the polymerase built. So let's try this out. The helicase, right, let's say, looks like so, right? It comes in and separates what's there. Then the polymerase, let's say, is just a glob like that, will then kind of hold up a little flag and lead the polymerase, we'll say just as an X, which then builds and comes along. Then the ligase, we'll just draw a square for the ligase, then comes along and follows all of them and glues it all together. This whole process starts at the origin. Now we need to describe what the 5' to 3' and the 3' to 5' is. So basically, we take this sugar right here and we're going to number the sugar on the corners, starting at 1 and going around. So it's going to be 1, 2, 3, 4, and then instead of going here, we're going to go here for 5. So that's the 5' to 3'. And then it's going to be the same thing over here on the sugar. We're just going to label that S for sugar. So then it's going to be 1, 2, 3, 4, and then 5. So then it's going to be the 3' to 5'. So then we're going to go back up to here and label this one. Or, we already have it on this one. I drew two diagrams for less cluster. Why this is important is because the polymerase can only go from the 5' to the 3'.